Hello everyone, welcome to our class. I hope you all are fine and doing good. Okay, so as all of the students have visited to chemistry laboratory and you have seen many of the solutions are kept in the chemistry lab, and there are many test tubes and some more like dropper. Okay, so student, tell me your like tell me your opinion. What do you understand by this chemistry lab? Why and this solution and this test tube and this dropper? Why we are talking about this? For that, we need to do one experiment. Okay, so for that, there is one activity in your textbook. You have to check that. Okay, so like that activity is related to acid base litmus solution test. Okay, so we will come to know that acid base litmus test. Okay, so first we will check the effect of litmus solution, and then we have to find out the basic, it's basic or acidic. Okay, so share your observation after doing the activity. Okay, like you see that it turn red when added an acidic solution and turn blue when added a basic solution. Okay, so here is the sample sample and effect of solution and basic or acidic. You have to find out. Okay, acidic. Okay, so here we will see that when we are adding red, then we can see that it's become acidic solution. So here red, then it's become acidic solution. So after that, when we add, okay, so when we add red when an acidic solution, then turn blue when added to a basic solution. Okay, so later on, let's let's you tell me what do you infer from this activity and what is the indicator used in this activity. So okay, so now we will come to know about the natural indicator litmus as natural dye. Okay, so today's our topic will be the litmus natural indicator litmus as a natural dye. Natural indicator litmus as a natural dye. Okay, so now tell me, define this define this litmus in your own word. So here we will come to know that litmus a natural indicator. Okay, so what is the litmus natural indicator here? Okay, so here litmus is used most commonly used natural indicator which is extracted from leeches. Okay, so it has a mauve color in distilled water. Okay, like when we put this in acid, then we can see that the acid turn blue, litmus paper red. Okay, and when we put base, then we can see base turn red litmus paper into blue. Okay, so these are shown in change the color of blue litmus to red, whereas the bees are bitter and change the color of the red litmus to blue. Okay, so here the litmus solution is a purple dye which is extracted from lichen and a plant belonging to the division Thalapoida, and it's commonly used an indicator. Okay, so when the litmus solution is neither acidic nor basic, its color is purple. These are called acid base indicator. So this is called acid base indicator. Okay, so I hope everyone is clear. Okay, so moving further, now tell me, you have to discuss about the following verbally and find the relation between them. Okay, like if I have the baking soda and water and vinegar, okay, so what will be the reaction here? So here we can see that red cabbage, the juice of red cabbage is originally purple in color, okay, and juice of red cabbage turn reddish with the acidic and turn greenish with base. Okay, so again I have one statement for you and you have to find out is it true or false. Like when a blue litmus paper is dipped into an acidic solution, then blue litmus paper turns red. Is it true or false? I hope everyone have listened the question and you will opt the correct answer here. Okay, so the statement is true. So okay, so moving further, okay, you have seen like onion in your home. Okay, so now you have to identify the concrete object and find out connection between from them. Like if I say you this is a onion, and after cutting the onion we cut into a pieces like this. Okay, like this, like this. Okay, this is the pieces of onion, and because we have chopped this onion, and this is the full big onion. Okay, so now you have to do one experiment, okay, related to the this. Okay, so here is olfactory indicator, and you have to look carefully and share your observation. Okay, so what is the observation here? Like you can see that on adding basic solution to onion strip, the smell of the onion is not detected and acidic solution does not eliminate the smell of the onion. Okay, so very good. So here what is the main ingredient used in this activity to test whether it is acid or base, and what is the name of Sole sensory nerve and convey the sense of smell. Okay, so here we will come to know the concept of olfactory indicator. Okay, so now you tell me what do you understand by olfactory indicators. So here olfactory indicator is that like substance which change their smell with mixed with acid or base are known as olfactory indicators. Like for example we have seen onion, clove. Okay, for onion we can see that in the form of acidic it will be the smell will be the retain smell, and base will be the losses its smell. Okay, so this type of indicator are used in laboratory to check whether a given solution is basic or an acidic solution by a process is known as olfactory titration. Okay, so the olfactory indicators have a characteristic odor. Okay, so this odor in olfactory indicator is change the acidic or basic solution and help us in detecting whether a given sample solution is acidic or basic in nature. Olfactory indicators are used to ensure the participation of visually impaired students in the laboratory. Okay, so coming to next, like we have seen like ice cream having the one type of cube of ice cream having the cream, and we have vanilla like flavored. Okay, and we are having rose like we are acidic and base in test tube, we put acid and base in test tube, like acid is HCl and NOH in base. Okay, then we have to find out the smell of vanilla vanishes with base, but its smell does not vanish with an acid. Okay, so here we use the vanilla to smell like we have found that the smell of vanilla vanishes with base, but its smell does not vanish with an acid. Okay, so I hope everyone is clear. So later I have one filling the blanks question, like those substances whose smell or odor change in acidic or base solution are called what? Are called what? Are called olfactory solution. Like you can say as indicator. Okay. So this will be the answer here. Okay. So moving further. Yes. Okay. So moving further, as you have seen that China rose and onion, turmeric powder. Okay. So as like we have seen that long, we have seen long also. Okay. So you have to identify the types of indicator in this. Okay. So okay. So here you have to observe like we are having three types of solution. Okay, I am writing here on whiteboard. Like first we have phenolphthalein, and second methyl orange, and third one is methyl red. Okay, you have to observe this carefully, these three solution, and you have to construct the meaning of this three solution. Okay, so here I hope you will find out the okay the concepts. So here we will come to know the concept of synthetic indicator. So you have to identify the concept of synthetic indicator. Okay, so let's define this synthesis indicator. Okay, in your own word. So we will explain about synthetic method. Okay, so here indicator that are synthesized in the laboratory are known as synthetic indicator. For example, we have seen that phenolphthalein and methyl orange. Okay, so this comes under the examples of synthetic indicator. Okay, so here phenolphthalein is an colorless liquid. It remains colorless with acid but turns into pink with a base. Like here we have seen like this is a acid pH. Okay, when we apply phenolphthalein, then we can see that it remains same only. And when we see base pH, okay, so here we can see that it's turned into pink color. So this will turn into a pink color. Into pink color. Okay, when we add in this in base pH phenolphthalein, then it will convert into a pink. Okay, so this is the process of. So this is the synthesis indicator. Okay, so here we will come to know about the statements like methyl, like methyl orange and is originally orange in color. It turn into the red with acid and turn into the yellow with base. When we put in methyl orange, when we put acid, then it's turned into pink, and when we put into the base the methyl orange, so it's converted to the yellow color. Okay, so here I have one question for you, like while performing an activity the levels produce phenolphthalein. Okay, so a pink color is observed in test tube. Okay, so you have to find out the observation. Okay, so let's recall our today's class. So in our today's class we have learned about types of indicator. Now let's summarize today's class. First we have discussed about litmus, litmus is the most commonly used natural indicator which is extracted from leeches. And second, we have discussed about olfactory indicators. Like in olfactory indicator, substance which change their smell when mixed with acid or base. So there, after that we have discussed about synthetic indicators. So what is the synthetic indicator? Like indicators that are synthesized in the laboratory. Okay, so later I have homework for you. Like you have to differentiate between natural and synthetic indicator. Give list of what color natural indicator: red cabbage leaves, colored petals of China rose, and litmus give in acidic and base medium. Note down the following in the table. Okay, I have given you the table. Like the second question is that why does a curry stand turn red with when it is washed with a detergent? Okay, so third question is that what is olfactory indicator and how it is used in the study of acidic and base? Take the example of any two indicator to support your answer. Okay, so last I have one activity. Like here in this activity we need red baggage, a glass of water, pen and cutting knife. For this instruction is given over here. Finally chop red cabbage, then take about glass of water in pan and boil it, and add the chopped cabbage to it. Allow the mixture to cool for a few minutes. When this liquid has cooled, strain it into clear glasses and discard the cabbage. Red cabbage juice will be obtained as a dark reddish purple solution as a natural indicator. This can be used. Okay, so here I have given table like indicator, substance, color in natural solution, and color in acidic solution, and color in basic solution. Okay, so you have to find out and tell me the after doing this activity what you will find out. Okay, so I hope you have written over this homework in your notebook and you will do this homework. And please come tomorrow. Okay, so we will discuss about this activity. Okay, so I hope everyone will do. So thank you everyone. Bye.